This lecture will cover analysis of variance, or ANOVA, which corresponds to Chapter 11 in the textbook I'm using now, which is Hyman's Stat. So far, we've learned, apart from descriptive statistics and graphing, which of course are very important because this includes measures of central tendency, sampling distributions, and measures of variability, right? The procedures we learned for testing hypotheses have included correlation, regression, z-tests, which I'm not actually including in this list because we usually don't use it, and t-tests, one sample, independent samples, and paired or related samples, t-tests. But what happens when we want to compare more than two means? Because so far we've compared two means with our two sample t-tests, but what about if we have more? So I'm going to do a brief review of the scientific method and experimental design here to lead up to what happens when we need to compare more than two means. So we've seen this before. The scientific method starts with generating questions and goes so forth and so on, right? We've been focusing on how to test predictions because this is what this course covers. But let's walk through an example. So generate questions. Here we can do it anyway. We can read the literature. We can look around ourselves. And this is something that I did some years back. So everyone says reading fiction is good for you. So you can start by wondering, is this really true, right? Did my third grade teacher make me keep a reading record for nothing? So then you look and review the literature or review what other researchers have already done and published. You'll find that lots of research shows a positive correlation between reading fiction and social cognition. Social cognition refers to the ability to understand the feelings, thoughts, and intentions of other people. It literally means thinking about others, right? Thinking about our interactions with others. So if we review the literature, we find that the more people read, the better they are at a test of social cognition. So then we might ask a question. If prior studies suggest that reading fiction versus nonfiction enhance social cognition, and this is true for experimental designs too, I started wondering, well, what about visual media? We spend hours and hours watching television. Could watching fiction have the same beneficial effect reading it does? So based on that question, I would develop some hypotheses, such as viewing fiction versus nonfiction TV shows will have the same effect that reading fiction does. In other words, viewing fiction will improve social cognition, or how people understand and relate to other people. Next, we would formulate some predictions. For example, people will perform better at a test of social cognition after viewing TV dramas than after viewing documentaries if we are correct in hypothesizing that viewing fiction improves social cognition. Next, we would design and implement a study. So to test this hypothesis, I designed an experiment, which, as your textbook defines it, is a deliberate manipulation of one variable to see whether corresponding changes in behavior result, allowing the determination of a cause and effect relationship. Only true experiments can allow us to make causal inferences. So to do this, I had to operationalize my variables. Operationalization means finding a specific definition of a variable that allows it to be measured. So for example, fiction and film was operationalized as viewing TV show pilots of award-winning drama. Social cognition was operationalized as the ability to recognize emotion in pictures of eyes. In an experiment, the independent variable is the one that is manipulated by the researcher. In our example, it's the types of TV, fiction versus nonfiction. The dependent variable is the one that is measured to test the effect of the manipulation, right? It represents the measurable response or behavior of the subject in the experiment. In our example, the dependent variable is social cognition. The independent variable here was the type of TV show. participants were randomly assigned to watch either the pilot of an award-winning drama or an episode of a documentary, such as Through the Wormhole, an award-winning show as well. So we began with a simple study where people were randomly assigned to view either award-winning dramas or documentaries, fiction versus nonfiction. The experimental group watched dramas. We used the people who had watched documentaries as a type of control group, right? Nonfiction. In an experiment, you must use random assignment to control for possible confounds. This is just the process of assigning subjects to experimental or control groups randomly, such that each subject has an equal chance of being in either group. And we talked about this before when I walked you through an experimental design with the example of the effect of cameras on test scores, right? In this example, our dependent variable was social cognition. It was operationalized as scores on the eyes test, or the reading the mind in the eyes test. In the eyes test, participants complete a test which has 36 items or questions that are photos of people's eyes, and it's multiple choice. They have to choose which of the words best describe the emotion in the eyes that the person is feeling, right? In this case, the emotion, the correct answer is playful. Right. So we had participants come into lab, watch television, and then take the eyes test. Then we had the data, we cleaned the data, and we could use it to test our predictions. Remember, the hypothesis was that viewing fiction versus nonfiction TV shows would improve social cognition. In other words, this is true. People who view award-winning dramas will score higher on the eyes test than those who view documentaries. This is a directional hypothesis, and we need to use a one-tailed test for it. Just to remind you, for an independent samples t-test, this is independent samples because they're different people, the null hypothesis is that mu1 minus mu2 is less than 0. The alternative, which corresponds to our research hypothesis, is that mu1 minus mu2 is greater than 0. The means in this case would be the mean for fiction minus the mean for documentary is going to be greater than 0, right? That is our hypothesis, because we expect scores on the eyes test for people who watch the award-winning drama to be higher than scores on the eyes test for people who watch the documentary. Here are the results of that analysis. You can see that the mean for people who watched drama was 24.55, and the mean for those who watched documentary was 27.28. So right now we check, are we right? Yes, people who watched drama had higher scores on the eyes test than people who watched documentary. Because we were right, we can go and look at the one-sided p-value. Right? It's 0.031. This is less than 0.05, so we can reject the null and conclude that people who watch award-winning dramas will be better at social cognition than those who watch documentaries, at least in the immediate aftermath. There's our one-sided p-value, and there's our point estimate of Cohen's d, so d equals 0.36. Finally, we get to draw inferences and say, it looks like a one-off exposure to award-winning drama improves people's ability to recognize emotions in people's eyes. And so we wrote this up and submitted it to a journal, Psychology. Okay, hang on a sec. Okay, so what happened? Well, it was reviewed and we got to revise and resubmit, which is a good thing. When you submit an academic article, you almost always have to revise it, which generally makes it much better. The journal editor wanted us to have a true control group, that is, a group of people who took the eyes test without exposure to any type of television. So just to review, independent variable groups are the experimental groups and the control groups. So experimental groups receive some treatment or are subjected to some things, such as watching TV, to the independent variable, right? In this example, they watch TV. They either watch fiction or nonfiction. The control group includes subjects who are not subjected to the independent variable in any form. They might receive a placebo if it's a medical trial. This controls for confounding variables and things such as experimenter bias, placebo effect as well. In our example, the control group did not watch any television. Our predictions is that people will perform better at a test of social cognition after viewing TV dramas and after viewing documentaries, and better than a control group. So we gathered the data with more people this time, and we tested our predictions. Our predictions, if-then statements. If watching good TV fiction improves social cognition, then participants in the experimental group who watched award-winning dramas will do better on the eyes test than those in the documentary and control groups. How do we test for mean differences between three groups? We can't use an independent samples t-test because there's three. We could do 3 tests, but we'll talk about why we don't want to do that. We used an ANOVA, right? And here is a figure that shows you the means. And it shows that, at least as far as we can tell, here we have on the y-axis the mean eyes test results, and on the x-axis we have the 3 groups, right? We can see that scores were higher for the fiction group compared with both the documentary and the control. We don't know, based on eyeing a chart, whether these were statistically significant. But it turns out they were, right? We can draw inferences. Fiction and film can improve social cognition, but then we might have more questions. We might want to repeat, replicate, and extend the study, right? So I'm going to stop this video now, and we'll do another one.